Welcome everyone to this MedPhysio tutorial on one of the most fascinating junctions in human physiology, the neuromuscular junction, where nerves and muscles meet to create movement. Think of this junction as the handshake between two systems, the nervous system which commands and the muscular system which obeys. At this microscopic interface, a burst of electrical energy from the neuron is converted into chemical language, and that chemical message awakens the muscle into mechanical motion. Every deliberate action, from writing your name to taking a deep breath, starts right here. The neuromuscular junction is where intention becomes motion and biology becomes behavior. In this lesson, we'll explore how this tiny synapse is built for speed and precision, how it translates signals flawlessly, and what happens when this process breaks down in disease or under drugs. Welcome to the neuromuscular junction, where physiology turns thoughts into action. Now, here's how our lesson flows. We begin with an introduction and structural overview. Here we see how the neuromuscular junction is built for precision. Then we move into the sequence of transmission, where the choreography of ions, vesicles, and receptors. Next comes electrical mechanical coupling, describing how sparks become strength. After that, we'll explore clinical and functional significance. Here, physiology meets medicine. And we'll close with exam strategies and a recap to tie every concept back to how you present it under exam conditions. By the end of this tutorial, you should not only recall facts, but truly understand and apply how the neuromuscular junction functions as a living bridge between thought and movement. You'll first be able to describe its structure. Then you'll explain the full sequence of transmission, step by step, from electrical ignition in the nerve terminal to mechanical contraction in the muscle fiber. Next, you correlate the electrical, chemical, and mechanical events that happen in perfect succession. You also discuss the clinical importance of disorders like that interrupt this junction, like Myasthenia gravis or botulinum toxin paralysis, to understand how small molecular changes can stop movement entirely. And finally, you'll apply these concepts in exams, learning how to phrase your understanding clearly and gain every possible mark. So let's zoom in to the neuromuscular junction. At the tip of the motor neuron lies a bulb-shaped axon terminal packed with hundreds of tiny vesicles filled with acetylcholine. Think of these vesicles as messenger capsules, each one carrying a signal that says contract now. Now, facing this terminal across the microscopic divide is the motor end plate. This specialized region of the muscle membrane has deep folds called junctional folds. These folds dramatically increase the surface area, allowing thousands of nicotinic acetylcholine receptors to line up directly opposite the nerve's release sites. That alignment guarantees that when acetylcholine is released, it has the shortest, most efficient path to its target. Now, between these two lies the synaptic cleft space, an incredibly narrow gap, only about 50 nanometers wide, filled with extracellular fluid and the enzyme acetylcholinesterase. This enzyme is the gatekeeper of the system and also the timekeeper. It destroys acetylcholine almost as soon as it arrives, ensuring that every muscle contraction begins and ends precisely on schedule. Now, surrounding the clefts is the basement membrane, a thin supporting layer that anchors both the nerve terminal and the muscle membrane in perfect alignment. It's also where acetylcholinesterase is embedded so that cleanup happens exactly where it's needed. Together, these structures create a communication point designed for speed, specificity, and control. Electrical information in, mechanical response out, with no crosstalk or delay. Now, when the question says describe the structure of the neuromuscular junction, always list the 4 key components. 1, the motor neuron terminal. 2, the synaptic cleft. 3, the motor end plate. And finally, the basement membrane. And state one function for each. For distinction answers, mention that acetylcholinesterase terminates transmission and that junctional folds amplify receptor density for rapid response. So now that we've seen how the neuromuscular junction is built, let's move on to how it works. That is, the sequence of transmission events that transform a neural impulse to muscular action. An action potential travels along the motor axon and invades the axon terminal. This depolarization opens the stage. The inside of the terminal briefly becomes positive at roughly plus 30 millivolts, causing a steep electrical gradient across the presynaptic membrane. Because this terminal lacks voltage-gated sodium channels for regeneration, the arriving impulse must trigger chemical release, and it does so through calcium. The arrival of the action potential causes depolarization, which activates the PQ type voltage gated calcium channels. The extracellular calcium concentration, about 1.2 millimole per liter, greatly exceeds the cytosolic level, so that the moment these channels open, there's an inrush of the calcium ion driven by both electrical and concentration gradients. These transient rise in intracellular calcium, that's roughly a few micromoles, is the critical trigger for exocytosis. Without calcium, no transmitter is ever released. The calcium binds to the vesicular sensor synaptotagmin, activating the SNARE protein complex. This complex is comprised of 3 protein molecules. 1, the SNAP25, then syntaxin, and synaptobrevin. This molecular machinery pulls the vesicle and the presynaptic membranes together, leading to the fusion and exocytosis of acetylcholine. Each vesicle releases about 10 raised to the power 4 acetylcholine molecules into the synaptic cleft. There is a single quantum of transmitter. The combined effect of many quanta creates measurable depolarization of the postsynaptic membrane. Now, within roughly 50 microseconds, the released acetylcholine diffuses across the synaptic cleft and binds to nicotinic acetylcholine receptors on the motor end plate. Each receptor is a ligand-gated ion channel composed of 5 subunits. Acetylcholine binds to 2 alpha subunits to open the central pore. The channel allows sodium ion influx and potassium ion efflux, with sodium ion predominating, producing a local depolarization called the end plate potential, that's EPP. Now, unlike the action potential, the EPP is graded and non-propagated, but when it reaches threshold around minus 55 millivolts, it activates adjacent voltage-gated sodium channels in the sarcolemma, initiating a self-propagating muscle action potential. The muscle action potential rapidly spreads along the sarcolemma and penetrates into the T-tubule system, ensuring synchronous excitation of the entire fiber. This electrical wave will soon trigger calcium release from the sarcoplasmic reticulum, linking excitation to contraction. We'll explore that very soon. Now, transmission must stop as precisely as it began. The enzyme acetylcholinesterase, anchored in the basal lamina of the cleft, hydrolyzes acetylcholine into acetate and choline within milliseconds. Choline is actively taken back into the presynaptic terminal by high-affinity sodium-dependent cotransporter to resynthesize acetylcholine using choline acetyltransferase. This recycling ensures that even with continuous firing, the supply of transmitter remains steady. The diagram below quickly summarizes everything that happens from the arrival of the action potential to the breakdown of acetylcholine after it's been released into the synaptic cleft. So now that we've seen how a nerve impulse crosses the neuromuscular junction, let's connect that electrical spark to the actual mechanical contraction of the muscle fiber, the process known as excitation contraction coupling. The sequence begins when the nerve action potential depolarizes the motor terminal, producing an end plate potential in the muscle membrane. If this local potential reaches threshold, it generates a muscle action potential that sweeps across the sarcolemma and dives down the transverse T-tubules. Within the T-tubule membrane lie voltage-sensitive dihydropyridine receptors, which are mechanically linked to ryanodine receptors on the sarcoplasmic reticulum. When the T-tubule depolarizes, the dihydropyridine receptors undergo conformational shift that opens the ryanodine channels, releasing a surge of calcium ion from the cytoplasmic reticulum into the cytosol. This sudden rise in intracellular calcium ion concentration binds troponin C on the thin filament. The binding displaces tropomyosin, exposing the myosin binding sites of actin. Now, the cross-bridge cycle begins. Myosin heads attach, pivot, and pull the actin filaments inward, producing tension and shortening the fiber. Let's integrate this as a summary. So the complete chain reads: the nerve action potential leads to an end plate potential, leads to a muscle action potential, leads to calcium release, leads to the cross bridge cycling, and after this you have contraction. Now let's take an exam perspective. When you're asked to explain excitation contraction coupling, always start with the from the neuromuscular junction. You can write it as such: the nerve action potential initiates an end plate potential that triggers a muscle action potential, leading to calcium release from the sarcoplasmic reticulum and subsequent actin-myosin interaction. It can also include the dihydropyridine and ryanodine linkage and the latency period. These 2 points distinguish an average answer from a first-class one. Now that we understand how perfectly the neuromuscular junction operates, let's explore what happens when this delicate balance is disturbed. Let's begin with Myasthenia gravis. Here, the body produces immunoglobulin antibodies against the nicotinic acetylcholine receptors at the motor end plate. With fewer receptors now available, the end plate potential generated by normal acetylcholine release becomes too small to reach threshold. This results in muscle weakness that worsens with repeated activity. That's a hallmark feature called fatigability. Treatment involves acetylcholinesterase inhibitors such as neostigmine, which allow acetylcholine to persist longer in the clefts and compensate for the receptor loss. Next is botulinum toxin, produced by Clostridium botulinum, which targets the presynaptic terminal. It cleaves the snare proteins, which are essential for vesicle fusion. Without these docking proteins, acetylcholine cannot be released and transmission stops. The outcome is flaccid paralysis, in which the muscles become limp because they never receive the signal to contract. Clinically, small controlled doses of botulinum toxin are used therapeutically to reduce spasticity or wrinkles. Then we have curare, the famous arrow poison from South America. Curare acts as a competitive antagonist at the nicotinic receptors. It occupies the acetylcholine binding sites without activating the channel, effectively blocking sodium influx and preventing depolarization. This produces reversible skeletal muscle paralysis, which is why such agents are used in anesthesia to induce muscle relaxation. However, note that the hard and smooth muscle remain unaffected. Their receptors are muscarinic and not nicotinic. Finally, neostigmine works by inhibiting acetylcholinesterase in the synaptic cleft. By preventing acetylcholine breakdown, it prolongs the availability of acetylcholine at the receptors, enhancing transmission. In Myasthenia gravis, this restores muscle strength. In contrast, excessive inhibition, as in organophosphate poisoning, can cause spastic paralysis due to continuous depolarization. So, acetylcholinesterase is both a safeguard and a vulnerability. Control its activity and it controls the junction's behavior. Now, when you are asked to discuss the clinical significance of the neuromuscular junction, structure your answer like this. State the normal function, rapid, one-to-one transmission between nerve and muscle. 2. Classify disturbances by site. Presynaptic, botulinum toxin. Synaptic cleft, acetylcholinesterase inhibitors. Postsynaptic, curare, myasthenia gravis. Explain the mechanism and resulting effects. It can be weakness or paralysis. Now end with a concluding line like this. Neuromuscular junction exemplifies physiological precision. Small molecular errors here lead to profound functional failure. So now let's move from physiology to performance. How to translate all this understanding into high scoring exam answers. Many students know the facts but lose marks because their presentation lacks sequence and integration. Here's how to answer neuromuscular junction questions to show understanding. Now, the NMJ appears in short answer questions as SAQs and occasionally as part of long essays or viva segments. Typical SAQs include 1. Describe transmission at the neuromuscular junction. Or they can come as differentiate between the end plate potential and the action potential. Now, both of these require physiological flow, not just random facts. Now, you can start with structure. Name the 4 components: the motor terminal, synaptic cleft, motor end plate, and the basement membrane. Then outline the sequence of events. Action potential arrives, leading to voltage gated opening of voltage gated calcium channels. This leads to release of acetylcholine and the binding to nicotinic receptors, the influx of sodium ion, the generation of an end plate potential, and which leads to the muscle action potential, and finally contraction. Remember, follow this with acetylcholine is degraded by acetylcholinesterase. Next, highlight the ionic bases. Calcium triggers release, very important. Sodium depolarizes and potassium repolarizes. Finally, state how acetylcholinesterase terminates the event. Never omit acetylcholinesterase. Examiners mark it as a critical detail, proving you understand, control, and reset mechanisms. Add numbers to show mastery. For example, the synaptic delay is about 0.5 milliseconds. The cleft width is roughly 50 nanometers. Each vesicle releases roughly 10 raised to the power 4 molecules of acetylcholine. Including these microfacts makes your script sound physiologically mature. Use these memory anchors to fix the concept permanently. ECM, electrical, chemical, mechanical. That's the full pathway of transmission and response. EPP starts it, AP spreads it. The end plate potential initiates the events, the action potential propagates it through the muscle fiber. These two short phrases summarize the entire junction in under 5 seconds. This is perfect for Viva answers. In essay questions, open with a definition. The neuromuscular junction is a specialized chemical synapse between a motor neuron and skeletal muscle fiber, where electrical excitation is converted into chemical transmission and then mechanical contraction. Then follow the flow diagram or bullet sequence we've practiced already. Always end with one integrative statement. This precise one-to-one transmission ensures that every nerve impulse produces a single coordinated muscle contraction. That's how you end on a distinction level. As we reach the end of this session, let's take a deep breath and trace the entire journey from thought to movement once again. Every voluntary movement begins with an electrical impulse in the motor cortex. That signal travels down the spinal cord, along the motor neuron, and reaches the axon terminal at the neuromuscular junction. Here, the impulse transforms into a chemical message through the release of acetylcholine. Acetylcholine diffuses across the synaptic clefts and binds to nicotinic receptors on the motor end plate, opening ion channels that allow sodium ion influx and generate the end plate potential. If this depolarization reaches threshold, a muscle action potential is born, spreading along the sarcolemma and diving deep into the T-tubules to trigger calcium release from the sarcoplasmic reticulum. Calcium then binds to troponin C, initiates cross-bridge cycling, and the muscle contracts, leading to generation of force, and movement begins. Finally, acetylcholinesterase ensures the signal ends cleanly, hydrolyzing acetylcholine and resetting the synapse within milliseconds. This ensures each impulse produces one precise twitch, not a second too long, not a millivolt too late. So, the sequence flows beautifully. Electrical excitation, chemical transmission, mechanical contraction, and finally, an enzymatic reset. That's the ECM continuum, the essence of your muscular physiology. When this system falters, so does movement. In Myasthenia gravis, receptors are destroyed. With botulinum toxin, vesicles can't release. Under curare, receptors are blocked. With neostigmine, we can rescue transmission by prolonging acetylcholine action. So let's take some time to do a mini activity. Draw, label, and explain. Take 1 minute now to sketch the neuromuscular junction. Label 5 key structures: the axon terminal, the synaptic cleft, the junctional folds with nicotinic receptors, acetylcholinesterase in the basement membrane, the direction of calcium and sodium flow. Then, beside each label, write its physiological role in one sentence. This simple exercise locks the concept in visual memory. That's the same strategy top students use before professional exams. The neuromuscular junction is more than a topic. It's the story of communication in its purest form, a conversation between nerve and muscle, conducted in ions and molecules. Every heartbeat, blink, and smile begins with these same sequence. Understanding it isn't just preparation for an exam, it's appreciation of how life moves.